Good evening, everyone. We are here for the session of our biological series. Today's topic is very interesting, Brain Living Powerhouse of Electricity by our speaker, Dr. Yogananda S.Markandeya. Currently, he is an assistant professor in Department of Biophysics from National Institute of Mental Health and Neuroscience, Bangaluru. Previously, he worked as an assistant scientist in University of Wisconsin, School of Medicine and Public Health Medicine, USA. He has done his postdoctoral research associate from University of Wisconsin. His PhD was in biophysics in NIMHANS, Bangalore, India. Master's was in chemistry from Kevempu University, Karnataka. Dr. Markandeya research interests are neuronal and cardiac channelopathies, patient-specific induced pluripotent stem cells, mitochondrial impairment in neurodegenerative disorders, and neurological disease. So with this introduction, I am pleased to begin our session and request Dr. Markandeya to start your presentation.
Thank you. So today I'll be presenting a talk on brain, and I'm very fascinated with this as a brain as a living powerhouse. The idea is this talk is made in such a way that a couple of our those who are new to neuroscience and the biology and those people who are away from neuroscience area. So I'm amalgamating all together, and also I'm adding some of our science too. Okay, so why did I say brain as a living powerhouse? So before that, it's just about my lab, how where and all we are working. So we are interested in neuronal disorders, especially looking at the epilepsy, aging, and psychiatric disorders. And main focus is on ion channels. And we'll be interested in studying the neuronal transmissions or a synaptic transmission, channelopathy, macromolecular signaling complex. And especially we are interested in calcium, sodium, and potassium, including glutamate receptors. And majorly we use, patch clamp techniques and optical imaging, as well as human-induced pluripotent stem cells. So here it is. As you can see that brain, if you connect a brain to a tiny lamp, it glows. So we can say that, it has a powerhouse, it generates electricity. So it's just like this, as we see in the image, like, it generates electricity. And you can see that, our brain is an, 3 pounds of average. The brain weight is average of an 3 pound, and it has, it's 3 pound jelly, and it has 2% of a body weight. It consumes 20% of the body energy, and it has totally 10⁻11 neurons. It makes 10⁻14 connections. So it does processing and information transmission and signaling. And it has a wide, the brain has been imaginarily divided into many lobes so that we can differentiate for our research and also the function aspects. So it is majorly divided into, frontal lobe, parietal lobe, and occipital lobe, and different lobes. Anyway, that's about a small introduction about the neuroanatomy. Then if you see, we people who are working with the electrophysiology, we see brain as this, as an electrical circuit. Here it is a telephone exchange circuit. You can see it's like a mind-boggling connections. Nobody knows where it is going. Only processors can tell us. On the right-hand side, you can see an as an artistic, signaling. And it depicts that, so much of signaling happens in a brain. It's a 10⁻11 neurons making 10⁻14 connection. It's like enormous connection. So how do neurons looks like when you take a rat brain and take it and then put it on a petri dish in a lab, and you can see these are the neurons. It looks like this. And when you stain them with a neuronal specific marker like MAP2, microtubule associated protein, so it shines a light. When you shine a light which attached to a fluorescence probe, and when you shine a light and see under the microscope, you can see like these neurons are green in shape, green in color, as you are seeing here. um, and Previously we were thinking neurons are alone, and later we get to know there are some glue like substance which is a counterpart of a neuron, and it also plays very important functions in neurophysiology. So here we stain them with a GFAP, which are staining for the astrocytes. They are the a side player we were thinking previously, and now we say that they also play a very important role along with the neurons in a brain. And if you want to see just a bright field image or a DIC image, you can see this. It's a cell soma, and these are connections. We can see them, networks, lot of arborization and things like that. So this is about how the neurons looks like. And if you go here is a cartoon of a neuron. You can see them. Neuron has a cell body, we call it cell soma, and then they have an axon, and then they have axonal tip, and these axon can go and connect to other neurons, to a dendrites, where it can make a synapse. So a signal can pass from one neuron to other neuron at that particular place. We call it as a synapse. And the neurons can receive their signal from a synapse, from uh, other axons through the dendrites. And right hand side we can see the other partners of neurons. They are the astrocytes, they are the glia. So the glia can be divided into three types majorly: the astrocytes, microglia, and oligodendrocytes. As we can see, oligodendrocytes make a wrapping around the neuron, which helps in conduction of a neuronal signaling. So it is just like this. If you take a electrical wire, and electrical copper wire will have a plastic wrap around on the coil, so that you can minimize the loss of electricity. So same way here, oligodendrocytes wrap around the neurons, and it helps in making insulations, and which helps signal reach faster to the end of other side of the neurons. And the microglia, they act as as an inflammatory markers, and they can help neurons. And also there are microglia, okay, oligodendrocytes and the astrocytes, which helps in so recycling the neurotransmitters and other nutrients. So there are majorly there are three different types of neurons. We can subdivide them. There are many neurons. Usually there are, like it is said that 10,000 different types we can see with respect to the morphology. I'm sorry, I can show you the Golgi stain, the different types of neurons, but anyway, cartoon is here with showing major different types of neurons. One is that multipolar neurons or an interneurons. You can see they are they have a cell body and the axons and dendrites. And another one is a motor neurons, and we know that motor neuron, as the name itself it suggests, it is responsible for a motor function, and it is the lengthiest neuron in the body. It can go up to meters. So you can see the cell body and then the axons. And in a motor neuron we can see that the oligodendrocytes or Schwann cells, Schwann cells, they wrap around and makes uh, a myelin sheath. And there are sensory neuron, which takes up the sensory elements like either a sound, smell, or a vision or anything to the central nervous system. So there is a sensory neuron, and the sensory neuron are look like this morphologically, and they are it kind of a bipolar in and structures. We can see a cell soma on either side it can go. So yeah, these are major type of neurons we see in a brain. And so how does these neurons interact between each others? So if, say for example, you can take in this cartoon, there are many neurons connected each other. You can see this neuron connecting to the this one and this one too, and they make a junction. So that junction is called as a synapse, where there are presynaptic, which is the signal is coming, the signal comes from one neuron, we call it as a pre, and the signal accepted by another neuron, that we call as a post neuron or a post synaptic neuron. So cartoon shows, this is a presynaptic neuron, and here is a post synaptic neuron. The signal reaches to the end of this presynaptic neuron, and as soon as signal reaches the channels, ion channels open, and especially the calcium enters, and the neurotransmitter release happens. So as soon as the neurotransmitter release happens, then the signal continue through the postsynaptic neuron. If you just see a synapse, how exactly it looks like in an EM, so we can see this is a chemical synapse, because neurotransmitters are released, that is the chemical. And there is another type of synapse which I'm not going to talk about. It's electrical signals. It is between two junction, and it's made up of connexins. So electrical molecule can go between each other. So this one is chemical synapse. We can see lot of vesicles, these vesicles filled with a neurotransmitter. As soon as signal comes in, it releases, and we can see this, the space between them is called as a synaptic cleft, or where there are neurotransmitters and ions can release, and they also localizes there. And in on the postsynaptic membrane, we can see very dense electron dense patches, because they have a lot of ion channels and other signaling molecule associated with that, and so that it can propagate downwards to the other neurons. So that is about a chemical synapse. Now let us see how these neurons generate electricity. So when you see this, when you look at the neuronal membrane and cardiac membrane, we call it as an excitable cells. And these membranes has acts as a semi-permeable membrane for in case as a case. You can see usually the neuron or in cardiac system, they have an extracellular and intracellular fluid. And when you look at the extracellular fluid, we see most of the time the sodium is higher and potassium is lower. It's in the extracellular I'm talking about, and the calcium is very high. It's 2 millimolar at extracellular space, and the magnesium is around the same 1 to 2 millimolar, and so what happens intracellular, sodium is always less and potassium is always higher. So it may be around, it varies in different cell types, and on an average we can consider like around 145 millimolar. So the idea is we can see these membranes are separating some of these charges. Say for example, here we talk about a sodium, the sodium ions. Sodium ions are having not only the positive charge on it, it also has a concentration difference. So now across the membrane, we can see there is a concentration difference, also charge difference. Now, because of this concentration of charge difference, we can call it as a potential difference. So this is our typical battery. An electrical circuit, you can show it as shown here, a positive plate and a negative. This is how we call it as a potential or a battery. Now, as we see that the charges are separated by these excitable membrane, so these acts as a charges which is either positive charge or a negative charge sitting on either side. So that is a charge deposition. So it acts as a capacitor. So there are some integral membrane proteins, we call it as ion channels, which opens or closes with respect to the potential difference across the membrane, or in other ways some ligands whenever it binds. So that means we consider them as a resistor. So now in a membrane we could see all the three. One is the potential difference as a battery, and other one is a capacitance and also a resistance. So now we have an RC circuit. So if you derive, and Atkin actually and Goldman, he derived based on Nernst equations to calculate the potential difference. This is given by E is equal to RT by ZF, and natural logarithm of ion concentration outside and inside, and you can put it for all the ions and summate it. As we know that major ions are sodium, potassium, and sodium, potassium, and chloride for the resting membrane potential. So if you substitute all the concentrations and the charges, we could get the resting membrane potential of a neuron. So these elements are governed by our physics laws like Ohm's law as well as the Kirchhoff laws. So now that there are, as I said, there are different types of ion channels. So these are the transmembrane protein. You can see here, some of them are opens and closes with respect to voltage difference. We call it as a voltage gated one. And the ligand gated, or when a ligand comes and binds, it opens or closes. So it opens, say, especially your excitatory, especially the glutamatergic neurons like NMDA kinase and GABAergic neuron, they are inhibitory neuron, so inhibitory receptors, and others are intracellular gated, like ligands from the intracellular itself, they can bind to the receptors and open and close. It's just like a gate. So these are especially the KATP channel and other one. So and the other one is the stretch activated, the mechanical sensor ion channel. Now let us see how these ion channels generate an action potential or a signal. We call it, this is with respect to a one single neuron we are talking about. Say left hand side, you can see an y-axis, it depicts membrane potential. So usually in neurons we see based on the ionic distribution that resting membrane potential is around minus 70 millivolts. And then as soon as there is a chemical release, neurotransmitter release happens, and the neuron takes up that and the signal pass through it, and as soon as it reaches to the another neuron, we can see that this will be at minus 70, and now there will be a small potential changes, and summation of potential changes at the axon lock makes it to reach around minus 55, where it reaches a threshold, and now it shoots up an action potential. It goes to plus 33 millivolts. So what happens during that, when it reaches the sodium ions or a sodium channels, the sodium ion enter through the sodium channels. So what happens, the sodium concentration is higher outside and lower inside the membrane. As it is from higher concentration to lower concentration it comes, and the inside the cell membrane, inside the cytosol, it's most of the time it's a negative because of amino acid, and they have a charges and proteins and other things. So what happens is that it's negative, and as it comes, we can see the y-axis, it goes to a more depolarized state, from higher polarized state to a depolarized state. When it starts reaching up to a 0 millivolt or so, what happens, the sodium ion starts slowly closing, or potassium ion start open slowly. And sodium channel has a property, it can open and close within 2 milliseconds. So this is a very fast gated ion channels. It closes within 2 milliseconds. It has an inactivation. So as soon as it reaches a 0, the potassium start opening, and at the same time the sodium channels start closing. So once the sodium channel closes, the potassium ion starts still opens. So potassium ions are open, and it can let the potassium ions from inside the cytosol, it can let it go outside. So what happens is that now the membrane it start repolarizing. That means from a positive to a negative it starts going down. So it started going to a repolarized conditions. So it, as we know, the sodium channel opens and closes very fast, and the potassium channels take some time to close. So as it takes a delay, so we can see it can go up to a minus 90 millivolts, and then slowly it comes back to a minus 70. That is the resting membrane potential. So now this process will happen. So this is how a single neuron can generate an electricity. Okay, now how do we measure these single neuron electrical activity? So we use a setup called patch clamp. Here you can see a microscope. It's here, it's an upright microscope. You can see here are the pre-amplifiers and amplifier. This is the amplifier, and then we have a micro manipulator to control fine tuning of a pipette. So here is a glass capillary. It's a 1.5 od outer diameter, 1.5 outer diameter, and 0.86 inner diameter. You can see there's a very fine capillary, and we can pull it into a very fine tip. It's around 1 mega ohm to 2 mega ohm bore size. That will put it into a preamplifier and maneuver in such a way that it should reach to uh, cell. And oh, okay, I'm trying to get that movie out there. Okay, all right, the movie is not playing anyway. So it goes to a neuron and it makes a seal, and then we cut open, we will just apply a small section and we open it, and now the cell is in contact with a intracellular moiety of a cell and the pipette. So now we can record the electrical activity of the neuron. So this is how we measure a single cell electrical activity. Here is just to give you an idea how we can see the difference between the different types of different age neurons. So we have cultured primary neurons. So it's from the rat neuron, cortical neurons. So they are of 6 to 8 days. We call it young neurons, and 22 to 25-day-old neurons. So when you patch them and record their electrical activity, we call it as EPSCs. So these EPSCs, we can see these are the excitatory potentials. We can see a small potential, and you can see, appreciate that amount of current we record. It is around 10 picoamps. So it's a very, very tiny current what we measure. So when you see there is a difference between young and old, we can, in terms of amplitude and also in terms of frequencies. So, and then we generated the action potential. Action potentials can be generated by just injecting uh, uh, electric current, or you can call it picoampere of current. So we can see that when you apply the similar amount of electric current to a young and old neuron, you can see that the younger neurons are firing a more action potential compared to an older neurons. So this can easily differentiate the physiology of two different types of neurons. And it can be the same way we can study a disease condition. Say just here is another example, right? Left hand side, I'm showing you a hippocampal neuronal culture. So these are the neurons taken out from a rat hippocampus, plated and cultured. And then what we did, we added CTZ, a drug which potentiates the glutamate receptors, and then it mimics the epilepsy in the human brain. So now when we measure the electrical activity of the single pyramidal neurons, we can see in one action potentials, we can see there are many. That means it's a burst firing, it's firing a lot. However, if you see a controlled one, we can see there is a one single action potential and the firing is very, very minimal. So this is a normal, and in epileptic condition we can see. This is how we can see in epilepsy uncontrolled firing electrical activity in neurons. Now that we know that we can record a single neuronal electrical activity in our laboratory, and we can study the neuronal dysfunctions and pathophysiology of it. Now, how do we study the higher order electric current? So people studied the electrical activity of the brain using electroencephalography. It's commonly known as EEG. So what happens is that the people put the electrodes on the scalp and measure the electrical activity. In the left side, you can see these are the electrodes which are on the scalp, and below that you can see a skull and the dura and pia mater and subarachnoid space, and then comes the neuron. So we can see these ensemble of neuron. When you put an electrode on the surface of the brain, you can get around 1 to 2 millivolts of current, but when you move on to the scalp, and you can get around 100 microvolt of and electrical impulses. So then what you do, you just connect it to an amplifier and you can record. Here I'm showing only one single electrode. In between the electrode and the scalp, we put some gel which gives you a better conductivity. So now this is how a simple electrical circuit which shows how to record electrical impulses from the scalp. And you can see this is a cartoon which is showing cap. This cap has many electrical electrodes, and it can be owned by a patient or a person. And these are below these electrodes, they put some of the gels so that for a better electrical conductivity, and now record it. So what happens is that when you connect it to amplifier and readout device, you can see the summation of non-propagating dendritic and somatic postsynaptic potentials. We can see it. So how do people usually use it in diagnostic? So you can see on the right hand side, all these electrodes are marked based on the different parts of the brain. So they have a definite system. They call it 10-20 electrode system. They have measured from nascent to 10%, and based on 20%, 20%, and then again to 10%. So they place these. Nowadays it comes easily because we don't have to mark it out. It comes with a cap. You just put it on it and connect it to the amplifier. So now what information does it gives? So it talks about the location wherever the electrodes are there. So it talks about that particular part of the brain activity. Say it can be in frontal lobe or parietal lobe, temporal lobe, and like that. And then amplitude. So we can also see the amplitude of the signal. So how big, how much it is. And then it also tells us what exactly it is. And morphology of it, whether it's a sinusoidal spike or things like that. And also we can look at the frequency. In the frequency, we divide them into multiple like delta, theta, alpha, and beta frequency. So delta leads to 0 to less than 4 hertz, and theta is between 4 to 8 hertz, alpha is between 8 to 13 hertz, and beta is again like it's a more than 13 hertz frequency. And then we also look at the rhythmicity, and then also we look at the continuity and clinical status, whether the person is asleep or awake, or seizures, or he's having a seizures or any other thing. So now just to give you an idea that how the brain activity happens and which are the wave functions we could see. So if you consider the beta waves, which is usually from 15 to 50 hertz, so these waves we can see they are very fast, at alert or working. And alpha wave, we can see it is between 9 to 14 hertz. It is usually relaxed or reflecting or things like that. That's the state of the mind. We can see the alpha waves. And theta waves we can see when a person is drowsy or ideally he's sitting or doing something. And then you can see the delta waves. It is very, very less. Its frequency, it is 2 to 3 hertz. So during a sleep or it's actually no dreaming, and only it's total asleep, a total sleep, a deep sleep you can call it. And then you can see the beta waves. The beta waves is between 15 to 30 hertz. It's usually, it's like during the REM sleep. REM sleep is like, rapid eye movement sleep, and also dreaming. So we can see on the right hand side, you can see a PET scan of images, which shows like, where the brain is very active and it's inactive. And in case of epilepsy, how do we identify? You can usually see that these are the waves we can see, when before the seizure onset, and during the epilepsy we can see that, or seizures we can see, it's a spiking wave pattern, and then after the epilepsy we can see, a different wave pattern. So using this we can identify and diagnose the uh, many different activities, like sleep, or a stroke, or a cancer, or epileptic conditions, or many other brain or neurodegenerative conditions. So this is a very useful tool to study, uh, brain activity especially. So what are the application of it? So we can monitor, cerebral blood flow. We can identify, seizure locus, and we can see, even when psychiatrists or other people, when they do hypnosis, so we can see how long and all the quality of it. And the advantage of EEG is that, it's totally non-invasive. You don't have to open a skull or a brain or any or head or scalp or anything. It's just totally non-invasive, and it's very direct, related to metabolism, and it can be used in adults and children and different clinical populations. And it's very, quite uh, it's not like MRI, it's louder or anything. It's very quiet thing, and it has a temporal resolution of 1 millisecond or less than that, and it's better than, fMRI too. So altogether, now I have showed you, you know sorry so brain generates electricity, and how we can measure electricity in terms of a single neuronal level, and also to a overall or an ensemble of neuronal level, and what are the advantages of it, and how we can study the disease phenomena of pathophysiology of disease using these two, techniques. So that's all. So if you guys have any question, I would like to take.
Thank you, Dr. Markandeya. Can you please unshare your slide?
Okay. Okay.
Okay. So these are our lab students. So is there any question from anyone? Yes, Prashant.
Ma'am, I have a question, ma'am. Good evening, sir.
Yeah, very good evening.
Thank you, sir. Thank you very much for this beautiful presentation, sir. Sir, I want to ask that when a person is in sleep and he is also dreaming, he is also watching dream, so it consumes the more power of brain.
Yeah.
As it shows that the hearts 15 to 30 when a person in dreaming condition. So how it affect the brain conditioning, and how is it relaxing? How it can disturb our relax during sleep? How dream can disturb?
So you are what I
Yes, sir. Is how dreams can disturb our sleep?
how dreams can disturb our sleep. That's your question.
Yes, sir.
Oh no, I did not say that. Did I say that sleep disturbs our no, dreams disturb our sleep?
Okay. Sir,
No,
means
okay. What I mean to say is, here the brain activity, it's a neuronal activity. Okay.
yes.
So even though we are sleeping, our neurons are not quiet, right? They are consuming energy, and they are producing electrical signals, and they are processing, correct?
Yes, sir.
Correct. So what I said is, so when you take during the sleep, or when you are dreaming, so what happens is that we would see a different pattern of frequencies or electrical signals. That is what I was saying. In a way, it's a rhythm. So when you're dreaming, you would see a 15 to 30 hertz signal. When you are asleep, you would see 2 to 3 hertz signals.
Okay, sir. Thank you, sir.
Any other question from anyone?
Good evening, sir.
Yeah.
I would like to ask you a question, that is there any change in electrical activity of neurons during a migraine attack?
I am sorry, I have not followed those migraine literature, so I have no, I don't have idea about that. Oh, yeah, that's
Okay,
an
sir.
interesting questions, but I haven't followed the migraine literature on this. But I know that, during the sleep it affects, and during epilepsy, yes, but during migraine, I'm not sure. Or have you ever come across those literature?
So yes, sir. I was recently reading a paper. There they showed that there is overexcitability in neurons in
Okay.
migraine.
Okay.
So
Okay. So okay, as if migraine excites the neuron, absolutely we can use this. As I shown you, in a single neuronal level, if there is any excitability, if there is a firing in a neuron, that should be contributing to the ensemble of neuron in particular area of the brain, which can show us through the EEG signals. Absolutely, in that case, definitely it can be used to detect. But I don't know how much. What is the uh, uh, what is the amplitude level of, it could be what is the voltage level, like it's micron volt especially, but I don't know how much it is. So in that case, I haven't followed anyway. But
okay, sir.
thank
Thank
you for
you.
that information.
Sir, do you know the application of electrophysiology for mind-body technique? Because recently people are performing meditation or yoga, and they are looking for the changes in the brain structures or brain activity. So, do you have anything to say about it? Because most of the people are rare from the yoga background. They want to know at this level.
Yeah, so in our institute, uh, Dr. Bindu Kuti, she has a good grant, and they are studying about, the patients, and also the yoga people who are doing yoga, and the patient who are subjected to yoga, and they have seen that, their epileptic seizures are going down, and they could see that, what is that, clinically the yoga is helping, and such a people are they brought into the laboratory, and they have recorded their EEG signals. Definitely they are seeing a difference in that, the pattern. And also, if many long time back, they also recorded some of these people. They also have a grant, Buddhist monks who meditate, and then they brought them here, and they record. And also they compared with the same similar age group people, which has, they haven't done any yoga. They could easily see the difference in EEG signals. And they say that, especially these alpha waves, they are more in those who perform yoga and meditation and different techniques. So they would, that's what is called as mindful activities. So that helps them, keep them a better mental health. So that is how they showed it. And in Wisconsin, Madison, I could see that some people, they were working, and they used to invite sometime Dalai Lama and his group, and they recorded, all these signals as an hyphenated techniques, like not just EEG signal, even other technique also they used, and they could easily show that, doing yoga meditation, and also people try to say that 1 minute yoga or something like that, so that can really help better mental health. So this is what I have come across.
Thank you, sir. I have another question. Do you have any experience with the electrophysiological part of iPSC-derived neurons? If you know that how much they are similar to the primary neuron, if we compare the electrophysiological aspects of these.
Okay, this is a great question. So I have worked with iPSC neurons, and I could see that. Okay, let me tell you this way: people are seeing, protein levels. They are seeing genetic level. They are saying that once you reprogram them and then differentiate into a neurons or in cardiomyocyte, these both are excitable cells, they could see their physiology more or less same, but still there are some differences. It's more or less it's the same. It's more or less, as I'm saying, it's very clear, it's more or less, hence they call it neuronal like, or cardiac myocyte like, ventricular myocyte like. They don't call it exactly neurons or exactly cardiomyocyte, because they are not exactly the human originated neuronal physiology. But as I said, they express calcium channels, they express potassium channel, their action potential more or less similar, but they have a delay time, lag time, and also, when you take it to a conditions at 37-degree, what happens to the neuron and the iPSC cells, they are quite, or I could say, like, still 30-40% or even 20% difference is there. So we cannot call it exactly the same, but they show all pathophysiology as well as physiological behavior. So that's how we can say it, they are neuronal like, not exactly neuron, again I'm saying neuronal like and cardiac like. So this is a problem with the iPSC cells so far we are having.
Right, and people are widely using iPSCs because they are patient derived, have some feature of patients. So,
Correct.
but we want to achieve the similarity index with the primary cell, directly isolated from them.
Correct.
So
Correct.
we can mimic the best model for our
Correct.
drugs or anything.
Yeah.
Thank you very much, sir. So is there any question from anyone?
Yes, ma'am. This is Kalyan.
Yes, Kalyan, please ask.
Yeah, Kalyan, tell me.
Yeah, sir. Actually, this slow wave sleep or NREM sleep is important for us, but uh, it is says that NREM sleep should be improved, right? And
Correct.
RAM sleep should be decreased. So means they basically those are undergone dream, so they should reduce their dream phase and NREM sleep should be improved.
Yeah,
But
yeah.
but during the dream there are so many changes happen. Means there is also requirement of dream is required. Means uh, if you look at the electrical signal, so RAM sleep also have some importance to perform our brain activity.
Correct.
So what do you think that RAM sleep is totally, suppose I don't have total RAM sleep, is it good or bad?
So okay, let me put it like this. So okay, so there are multiple aspects. So we need all these activity in a controlled level, right? For a healthy brain, for the relaxed brain to work. So if something goes, see, when we are measuring all these things, REM sleep or anything, these are all physiological activity, correct, of a brain, right? So what happens is that we cannot say that, how do you say that this is higher, this is lower, and this is, a minimal, or so you cannot measure it, right? That's the first thing, because that's a mental activity, right? That's a brain activity. We don't have anything, because each brain or each person is individual, is separate. He require a different amount of, relaxation and things. Like some people can take, 1 hour of sleep and they can work. Some people need 6 to 8 hours, or some people can take in between, like, 1 minute of relaxation, they can work. So its physiology is different from person to person, brain to brain. Now when we talk about like which one is higher, which one is good, but this one also is required, like REM or non-REM or something. The idea is, so brain needs a rest. So like for its own, neuronal, what do you call it as, synapse formation or things like that, and all those things. The reason is, so when you're in REM sleep, it has a more activity, it consumes more energy, and the electrical impulses are so high that, our brain still needs more rest. So that way, it we say that, we should have a less of REM sleep, and we should have a higher of, non-REM sleep. But now again, you said even you need more of activity, right? So um, again, it all depends on a person, and also it depends on, the physiology of that brain activity. So we cannot say that which one is, it should be more or less or higher. So that's the problem with that brain activity.
Thank you, sir.
Any question from anyone? Thank you, Dr. Markandeya, for a wonderful presentation on the brain structure, neuronal structure, and their function and techniques which we can use to measure that. Thank you.